comes from Minnesota. And this is when I realized that Fargo is not in Minnesota, it's actually in North Dakota, but it's right there, and the movie is filmed mostly in Minnesota. But when I ask people in my lab what do they think when they say Lauren, that's some student in my lab, Megan, said Fargo. So she comes from Minnesota. And after that, she did her undergraduate degree at Stanford, and then she did some undergraduate research at the Hopkins Marine Station, where she worked with George Somero. For those of you who don't know who George Somero is, then he's basically one of the top people in comparative biochemistry. And George wrote me an email saying that Lauren had been one of the best undergraduates working for him. So I figured that if that was good for George Somero, Lauren should probably be good for me as well. And apparently Lauren did really well. She got a medal for excellence for her research. And after that, now what? So that's when she moved to Scripps. And joined my lab. It's like lots of pictures. I'm going to point out a couple of things. And number one, apparently in my lab we like to dress up in costumes a lot. The other thing that here I want to point out is like here Lauren is with Megan and Janae, all of which recently graduated this year. So Lauren hopefully will be the third one now. But the most impressive thing here is look at here Lauren dressed up as Hocus Pocus. It's like one of the best costumes I've ever seen. Originally, Lauren came with an NSF IGERT fellowship, which is a fellowship from the NSF for interdisciplinary research, and she did a lot of work on those topics. Some of that truly impressive, such as creating or leading interdisciplinary forums and the Ocean Scientists for Informed Policy, and also she spoke at the United Nations. And eventually we said, okay, that's great, Lauren, but we'll have to do some research. And she did research. She did really a lot of really good research. She started by looking at coral biochemistry and oxygen dynamics in corals. And the story behind this is that Lauren was originally interested in how corals might deal with ocean acidification from an energetics perspective. And the problem there is that we really don't know anything about coral energy metabolism. We know very, very little. So we basically made a deal, and it's, okay, what about this, Lauren? What if we will try to elucidate or discover these novel pathways in corals, and then if you have enough time, you can use that to look at corals in the field or to some other environmental source of stress. She started by looking at oxygen metabolism just in the normal corals during the day and night. As she's going to explain, she discovered a new pathway for creating energy, and then she finally was able to also do some research in the field in Panama at the Bocas del Toro research station from the Smithsonian. The first year in a project in which she was helping David Klein and myself in another project that was related to what she did. But then the second year she got a Stride fellowship, and she went there for 7 months, 6 months, in which most of that she was by herself. And the things here, again, two things I want to point out, three things. One, that that research allowed her to combine field experiments with laboratory experiments, which it's not very common. Also, like, just want to point out to Rubia, the dog that she adopted in Panama. And the third thing is this thing, which is a sloth. And believe me, I've never seen anybody more excited to see a sloth ever, or to see anything, not even a sloth. The first time that Lauren saw a sloth, she was weeping for like 20 minutes. Which I think is great, but I could not resist putting the sloth. Yeah, so now, after when she finishes her degree, already Lauren has secured a CNAUS fellowship to go and do a 1-year internship at DC. So looking forward to you helping drain the swamp. But yeah, just to leave you with the thing with Lauren's thesis, I really want to stress how important is some of the research that Lauren did, because she discovered a new pathway by which corals can make energy when there's little oxygen. And for corals, that is, for example, when corals are bleached or even at night. And just by knowing how corals, these new metabolic pathways in coral, that allows for further research in the environment to try to determine which species might be more or less resilient to climate change. and things like that. And also, this truly can provide new basic biological information about corals that is important for management and conservation. So yeah, it was 5 minutes, it's not too bad. So.
Hopefully I passed this part of the test. All right, well, thank you all so much for being here. Thank you for that wonderful introduction. Martine wasn't too embarrassing. And I'd also, in particular, like to thank all of my committee members for being here. Martine, my advisor, Dimitri, Davey, Horst, Bill, and Peter. And I'm excited to tell you today about the last 5 years of my research here at Scripps on the metabolic physiology of healthy and bleached corals. There we go. And to provide an outline of what my talk today will be, I'll first start out with providing a general introduction on corals and metabolism. This first part will be on the metabolism of healthy corals, and for this I draw on results from 2 of my data chapters on the dynamic oxygen microenvironment of corals, identification of strombine as the main end product of anaerobic metabolism, and my 5th chapter on detecting diel patterns in coral metabolism using untargeted proteomic and metabolomic approaches. And building upon our findings from the first part, we'll be delving into the metabolism of bleached corals. And I'll be using results from my 3rd chapter, which was doing a thermal bleaching experiment in Panama, and on these 2 important Caribbean species, Agrostia agaricides and Orbicella franksi. And I'm also throwing in a 3rd important coral species, Acropora cervicornis, as a bonus. And I also did some work on the effects of menthol-induced bleaching on coral physiology that unfortunately I won't have time to discuss here, but happy to give you a copy of my thesis, or we can talk more about it after. And then finally we'll go into some conclusions from my findings. I'd like to start out by delving into what motivated me to pursue this line of research at Scripps. And Martine provided a bit of an overview that I came here really interested in studying the effects of climate change and ocean acidification on corals. And that's because, to me, corals are absolutely beautiful and spectacular. But beyond that, what fascinates me is that this super thin-tissued animal is able to construct these massive reef structures that host some of the highest biodiversity levels on Earth, earning them their nickname, the rainforest of the sea. And we know that the high productivity of rates of corals are what hosts and support these high levels of biodiversity. But when I started my research here, I realized that we know essentially nothing about coral metabolism. And for me, I was interested in coral metabolism coming from a background of doing organismal physiology and knowing how important metabolism, or the set of biochemical pathways that produce energy, are to sustaining life. Thus, I decided to go into studies of basic coral biology focusing on metabolic pathways. And this might be a flashback to Bio 101 for many of you, but I think it's important to first establish that we're all on the same page with cellular metabolism and the importance of oxygen in governing the cellular metabolic pathways utilized in the cell. So it's also maybe basic to say that oxygen is obviously important for sustaining life. But this is important because with oxygen, cellular metabolism in the mitochondria can produce up to 38 moles of ATP per mole glucose oxidized. And here ATP is essentially the cell's main energy currency. In comparison to when there's not enough oxygen available, anaerobic metabolic pathways are about 19 times less efficient at producing energy. So clearly there's an important consequence of oxygen availability to the metabolic pathways utilized by the cell. And what do we know about corals? Well, we know that they're animals and that these pathways are generally well conserved between animals. But surprisingly, very little is known about coral aerobic or anaerobic metabolism. In particular, we didn't even know what anaerobic metabolic pathway corals have. But we do know from studies in other marine invertebrates that they can produce a number of these anaerobic endpoints. We all are familiar with lactate, which gives us the sensation of muscle burning when we're exercising. But marine invertebrates have evolved a number of different options, and I'm going to highlight a few of them for you that are functionally analogous to lactate. So we've got octopine, which is produced by octopus and sea anemones. Strombine is made by blue mussels, and elanopine is made by scallops. And marine invertebrates tend to produce these under shorter-term bouts of hypoxia. But in addition, when there's longer-term hypoxia in the cell, this can lead to the production of succinate and propionate in the mitochondria. We also know that, or while we don't know what the anaerobic pathway in corals is, we do know that central to coral metabolism is this energetically significant relationship between the coral animal host and its photosynthetic algae symbiont called Symbiodinium. And these Symbiodinium live in the gastrodermal, or two internal tissue layers of the coral polyp. And they're the main internal supply of both sugars and oxygen for corals, which are both key substrates for aerobic metabolism. We also know that this symbiosis, which is the basis of the productivity of corals, drives major fluctuations in the conditions in the seawater surrounding coral reefs. In addition to there being a lot of variability in other factors on reefs, such as light availability and temperature. And here we see that both pH and dissolved oxygen are much higher during the day than at night on coral reefs. And this is due to cycles of net production during the day and net consumption at night. Beyond this, what's even more relevant to the coral animal and its physiology is what's happening in what's called its diffusive boundary layer, which is the microenvironment that immediately surrounds corals. And in this layer, it's been found through some short-term studies using microelectrodes placed on the coral tissues that in the light, coral tissues are very hyperoxic or high in oxygen, whereas in the darkness, we see that they're very low in oxygen, reaching hypoxic or even anoxic, completely oxygenless conditions. So these findings would suggest that the cycles of high oxygen during the day for corals would support aerobic metabolism, and the low oxygen at night for corals would support anaerobic metabolism. And this leads me to one of my first main questions of my PhD, which was characterizing the oxygen microenvironment of corals throughout the day and the night to understand whether hyperoxia is sustained during the day and hypoxia is sustained at night, in addition to linking this to the consequences to aerobic and anaerobic metabolism. This is central to figuring out how much energy corals can produce for essential biological processes like calcification, growth, and reproduction. And then after studying this, I wanted to take a more global overview picture in studying day-night changes in proteins and metabolites related to metabolism. For these studies that were on the diel changes in coral metabolism, this required creating a really stable environment for these studies. So to do this experiment, I conducted a controlled aquarium study right here at SIO, where using the coral species Acropora youngi, I measured the oxygen concentrations on the semisarc tissue, which is the space connecting the polyps, over consecutive days and nights across multiple genetically identical branches to characterize this oxygen microenvironment. And I controlled for things like light, flow, and water temperature, which can all influence the oxygen environment. And then after getting all of this oxygen data, I sampled the corals at 6 time points over 24 hours and measured indicators of aerobic and anaerobic metabolism on them. Jumping into the oxygen microenvironment results, I found that like the previous short-term studies, there was average hyperoxia. We're looking at oxygen concentration here on the coral semisarc tissue, average hyperoxia during the day, and average near hypoxia or hypoxia at night in the tissues. And this is in comparison to the average conditions in the seawater surrounding the corals. Here I'm putting up a reference of what the commonly used threshold for hypoxia is to get a sense of just how low these oxygen levels are. Now this was really cool, but I would say what's even cooler is seeing what happened to each individual coral. And here we're showing the same data, but represented as individual coral branches, their oxygen concentration over day and night. And a couple of things stand out. One is that there's a lot of inter-individual variability, but there's also a lot of intra-individual variability in the oxygen microenvironment. So this has implications for levels of aerobic and anaerobic metabolism, which are driven by oxygen conditions. And to approach the question of how was aerobic and anaerobic metabolism in corals affected by these oxygen dynamics, I chose to focus on measuring activities of key enzymes involved in aerobic and anaerobic pathways. So one of our main aerobic pathways in the cell is the TCA cycle, and citrate synthase, or CS, is the first and rate-limiting step of this pathway. So measuring activity of CS is an indication of the total capacity of this pathway to produce energy aerobically. In addition, this enzyme, malate dehydrogenase, or MDH, is also measured often in combination with CS as an indicator of overall aerobic capacity. But I want to point out that MDH is also involved in a number of other metabolic pathways, which might become important later on. And then, as I mentioned, clearly we still don't know at this point what anaerobic metabolic pathway corals have. But, and here's where I felt like my thunder was stolen just a little bit during my PhD, but just last year, there was another grad student who actually was studying measuring enzyme activities in corals and detected activity of 2 anaerobic enzymes, strombine dehydrogenase and allanipine dehydrogenase. So I'd like to thank Murphy and Richmond for giving me the hypothesis of measuring these 2 enzymes as well. In addition to, I looked for activity of other key anaerobic enzymes. And what I found looking at the enzyme activities of corals sampled over the day and night is that in general we don't see huge changes in the CS or MDH activity in these corals during day or night. But in fact there was a significant increase in CS activity at night, driven by this slow ramp up of CS activity. And at this point it's important to talk about what was this assay measuring. And enzyme activity measures the maximum potential for energy to be made through these pathways. So this does not actually give us a sense of the instantaneous rate of aerobic or anaerobic metabolism, but rather just an idea of how much can be produced. Because otherwise it seems somewhat surprising that given the low oxygen at night, they would be producing more energy aerobically. In addition, there was much higher activity of MDH. If you look at the y-axis scale over here, about 100-fold higher MDH activity than CS, indicating that the other metabolic pathways this enzyme is involved in, such as gluconeogenesis, the malate aspartate shuttle, or the glycolate cycle, may also be important energy pathways in corals that require further studies. Now, in my studies of enzyme activity of anaerobic enzymes, I only detected strombine dehydrogenase, or SDH activity, suggesting that this is the main anaerobic pathway in corals. And again, we did not find any significant differences between day and night. However, to be absolutely certain of which anaerobic pathway corals are utilizing, you have to measure the actual production of the end product, so in this case, strombine, because these enzymes, enzyme assays only measure capacity and not instantaneous rates. So in order to confirm a anaerobic metabolic pathway in corals, I utilized highly sensitive liquid chromatography and mass spectrometry-based techniques, which allow us to, alongside the use of authentic standards of these compounds, measure the potential production of them in corals. And this work was done with Bill Webb at the Scripps Research Institute up the road. So I'd like to thank him for developing the LCMS methods that produce these results. And in order to confidently identify a compound in a sample, you need to meet several criteria. The first of which is that there's a similar retention time of the compound of interest. And that's what these two graphs on the left are showing. These are called extracted ion chromatograms. And first we see the chromatogram of strombine and the retention time of it. So in our coral sample, we're looking for something with the same retention time as our authentic standard, which we found within a small degree of error. The second criteria is having very similar fragmentation patterns, which signifies the molecular structure of the compound. And here for strombine, we see these main ionic peaks, which also appear in the coral sample. While these results heavily suggest that strombine is in the coral, because of slight differences in the relative abundances of the ions and the retention time, I decided it was appropriate to do further experiments to absolutely be confident of this pathway. And so to do this, I went back to the blue mussel, which again is well known to produce strombine, and it does this under conditions of anoxia. And I'd like to thank Dr. Youngjuan Su on Upper Campus here at UCSD for developing these LCMS methods. And we chose to develop some faster and cheaper methods in order to reproduce these more in the future. And here again, we see strombine, the chromatogram, and the fragmentation pattern. And as we would expect in normoxic mussels, which are known to not produce strombine, we don't see anything that looks like strombine. However, mussels that were exposed to anoxic conditions for 12 hours do show a large peak corresponding to strombine in addition to the same ions. Now, once again, we see this shift in retention time, which has an actual well-defined name called the matrix effect. And I like to think of Neo pushing the chromatogram to the left, but in fact, there's a much fancier explanation for this. So here we're showing that mussels produce strombine and that it also has a matrix effect. And to prove that the strombine in the coral samples also exhibits a matrix effect, we did a subsequent experiment using these same fast LCMS methods, where we spiked pure strombine into a coral sample and again witnessed that there was a retention time shift. So I think that in combination, all of these LCMS experiments confirm the presence of strombine in corals and indicated that it is the main anaerobic end product. And next, I was interested in quantifying the amount of strombine over the day-night cycle as an indication of the instantaneous rates of anaerobic metabolism. So to do this, we used even more and fancier LCMS techniques and in combination with multiple reaction monitoring, which is a quantification technique. And while comparing day to night strombin levels in corals, there's no significant difference, it becomes really apparent when looking at these individual time points that there are these two major peaks in strombin production. In addition, we detected the same pattern for this other opene called elanipene, but if you notice, there's about 100-fold less elanipene than strombin in the corals, again suggesting that, wow, this might be an anaerobic end product. Strombin is the main end product. And I was obviously interested in, so what is it about these two time points? And again, we can look back to the blue mussel, which also shows a pattern of two peaks in strombin production. One happens during anoxic exposure, which is happening here. And the second, even bigger peak in strombin production happens when these anoxic mussels are allowed to recover in normoxic conditions. And this is analogous to our two situations of the first peak in strombin occurring at night when hypoxia is first initiated from net consumption, and then again the peak during the beginning of aerobic recovery when hyperoxia returns from net production. So I would suggest that strombin production plays these dual energetic roles in both mussels and in corals in supplying energy both under anaerobic conditions and under highly energetically demanding aerobic conditions, which is termed repaying a post-hypoxic oxygen debt. And this is again showing the analogy between the two time points. So after I had found that strombin appears to be the main anaerobic pathway in corals, I was interested in backing up for a moment and saying what other changes to coral metabolism could be occurring day to night. Because clearly the strombin study suggests that there are these peaks in anaerobic metabolism. And for this question, I took two approaches where I used untargeted proteomics, which is basically taking our coral samples from the day and night, extracting out the proteins, separating them out on two-dimensional gels, and then digesting them into peptides, which we can identify through throwing them onto a mass spectrometer and comparing the sequences to known protein sequences. And I'd like to thank Marcus Uzo from Cal Poly for helping train me in these methods. And while this was a lot of work and it was really exciting because it was one of the first untargeted proteomic studies in corals, I found that the results weren't as interesting as I had hoped. But that sometimes happens with research. So I'm showing you here two heat maps of the proteins that change significantly between day and night. And what you might notice overall is that while there's some patches of yellow which indicate upregulation and patches of blue which indicate downregulation of protein, there were no clear patterns of a diel pattern in protein expression. We were able to identify 574 protein spots and identify 20% of these proteins, which is a fairly decent number considering the lack of genomic information for corals. But also, as luck would have it, using such untargeted approaches, sometimes you don't find what you might be looking for. And in this case, we did not actually detect CS, MDH, or SDH proteins in the coral proteome. However, what I am showing here is that we did find other proteins involved in aerobic and anaerobic metabolism in corals. However, there were no clear patterns in what was happening. Overall, the findings from our proteomic study suggest that while there might be some protein abundances that are changing day and night, any sort of metabolic regulation for corals probably isn't happening largely on this scale, but instead on a post-translational scale. Next, based on the findings of not much protein abundance changes between day and night, I wanted to look for changes that could be detectable on the metabolite or small molecule scale, which is a better indication of the fluxes through these pathways. And for this approach, I again looked to the Scripps Research Institute up the road, where we took an untargeted approach of looking for every ionic feature detectable using LCMS. And then with mapping algorithms in this online program, we were able to predict the significant metabolites, and then I mapped them onto known aerobic and anaerobic pathways. And again, to give a global picture of what happened, we detected a lot of ionic features, and only around 2% of these were significantly different between day and night, but this still represents a much higher number than of the proteins. And this is a nice just visual representation called the cloud plot, showing those ionic features that were upregulated at night and the ionic features that were downregulated at night, suggesting there was much more downregulation than upregulation. And based on the significant predicted metabolites, I mapped them onto aerobic and anaerobic pathways. And what we're looking at here is the significant predicted metabolites that were downregulated at night in red, and the ones that were significantly upregulated at night in green. And here we have our aerobic TCA cycle and some other anaerobic pathways that I haven't spoken much about yet, the glycosylate cycle and succinate propionate production, in addition to AMP de novo biosynthesis, which is a precursor for ATP. So what I would like the takeaway of this this mapping to be is that while we are unable to identify these metabolites down to a point, there's power in putting predicted metabolites onto known pathways and seeing that overall there's a pattern of downregulation in red of aerobic metabolism and upregulation in green of anaerobic. So this suggests, based on the metabolomics results, that while protein abundances weren't significantly changing, there do appear to be significant changes in metabolite levels, again suggesting that any metabolic regulation happening day to night in corals is probably happening in terms of changing the fluxes through the pathways. And so I know this was a long part one, and part two will be a little bit shorter, but to summarize what we found on the metabolism of healthy corals. So in part one, we've established that the coral microenvironment is generally hyperoxic during the day and nearly hypoxic throughout the night. And while this does not seem to affect the metabolic capacity of corals day to night, through looking at actual levels of anaerobic end products, it appears that anaerobic metabolism is peaking at the beginning of the night and again at the beginning of the day. And finally, that changes in daily metabolism are most likely regulated on the level of metabolites as opposed to on protein expression levels. Okay. So moving into part two, where we get to actually dive underwater. I was really grateful to perform all of those experiments here at Scripps, and I didn't really think that I'd have the opportunity to apply my research of basic biology to the field, but it was really a joy to get to spend some time in Panama at the Bocas del Toro Research Station of the Smithsonian Tropical Research Institute. And the objectives of this part of my research were to study the effects of coral bleaching on the oxygen microenvironment and metabolism of corals, utilizing some of the techniques I optimized in the first part. And in addition, because it's difficult to extrapolate lab findings to the field, I think it's important to, when we're ready, apply our findings to more ecologically relevant field and lab studies. And this is what the outdoor experimental wet lab at the STRI station looked like. And as luck would have it, while I was hoping to catch a natural bleaching event when I was down in Panama, I didn't, which is good for the corals, bad for me. So I conducted most of my experiments in these wet labs, which at least utilize natural light and seawater taken from off of a coral reef. So now to give a brief overview of coral bleaching. So coral bleaching is this major phenomenon occurring around the world for coral reefs, and it's mostly caused by elevated temperatures, which when temperatures rise, cause a breakdown in the symbiotic relationship between the symbiodinium and the coral, causing their ultimate expulsion. And this makes corals appear a ghostly white color. At this point, they're not dead, but they are very susceptible to disease and mortality. There are many things that we don't know about coral bleaching, such as the exact cellular mechanisms by which this symbiotic breakdown occurs, as well as what are the consequences of coral bleaching to the oxygen microenvironment and metabolism of corals, which, as you probably guessed, is what I was interested in studying. So why bleaching in Bocas del Toro, Panama? Well, the Caribbean has been particularly hard hit by these bleaching events due to rising sea surface temperatures and a number of other stressors occurring specifically in the Caribbean. This has resulted in some mass bleaching events in 2005, 2010, and 2015. And since I didn't catch a natural bleaching event while I was there, I conducted 2 thermal bleaching experiments in that wet lab. And I made measurements of the oxygen microenvironment. In addition, I measured rates of photosynthesis and respiration throughout the bleaching process, and I measured enzyme activities in the corals once they were fully bleached in comparison to control unbleached corals. And for these studies, I used 3 common Caribbean reef building species, a Cropper cervicornis, which used to be the main reef builder in the Caribbean, but has started to decline due to its sensitivity to stress. It also is known to have low recovery rates from stress. I also use Agaricea garicides, which is a foliose coral, and it's also known to be sensitive to stress, but it seems to have some higher recovery rates than a Cropper cervicornis. And then finally, to choose not only a different coral morphology of a mounting coral, but also one with a different susceptibility to stress, I used Orbicella franksi, which seems to bleach less readily and also have higher recovery rates in comparison to these other species. So during the first bleaching experiment, where I compared Agaricea garicides and Orbicella franksi, I monitored bleaching throughout the course of a thermal stress, which it took around 6 to 7 weeks to induce bleaching for both species at around 31 degrees Celsius, which was about 3 degrees above the ambient temperatures. And so during the course of this, I took pictures using color cards to monitor the levels of bleaching, and which can be seen here through the color score. So I'd like to draw your attention to the differences between Agaricea and Orbicella. Agaricea tended to bleach a bit faster on average than Orbicella, in addition to bleaching at the same rate, while Orbicella, some of them took much longer to bleach and had much more variable bleaching rates. And this also seems to match field observations. But during the course of the bleaching experiment, I was interested in studying what was happening to the oxygen microenvironment, as well as to photosynthesis and respiration. So I used the oxygen microsensors at 3 time points during the course of the bleaching. Initially, before bleaching began, so in unbleached corals, around the partial bleaching stage, and again when corals were fully bleached. While I monitored coral bleaching through looking at changes in coral color, to be absolutely sure of how bleaching occurred, it's best to measure densities of the Symbiodinium cells standardized to the coral surface area, as well as to the concentration of chlorophyll A over the surface area, which is the main photosynthetic pigment in corals. And I'd like to give a shout out to my great assistant, Cameron Hasibi, for doing much of this lab analysis here back at Scripps for these figures and for a few others. And what we found was that yes, coral bleaching occurred by significant reduction in both the Symbiodinium densities as well as the amount of chlorophyll for both species. So here we've just confirmed that bleaching occurred. And next, looking at the oxygen microenvironment of the corals throughout the course of bleaching, what I found was that in healthy unbleached corals, they were very similar to the healthy corals I measured here at Scripps, where their oxygen environment was very hyperoxic in comparison to the surrounding seawater. Interestingly, as corals bleached, they lost oxygen concentrations, and partially bleached corals mimicked the conditions in the surrounding seawater. this was really interesting to find that fully bleached corals actually were completely undersaturated with respect to oxygen during both the light and the dark. And now this is, these are measurements made under the light, and these are measurements made in the dark, as we also expected to find healthy corals had significantly lower oxygen levels in the dark from net respiration occurring. But you'll see that partially and fully bleached corals had a slight relief of nighttime hypoxia. And this is probably because when corals are bleaching out their symbiodinium, there are fewer things there to add to the respiration signal at night. But the main takeaway from this result is that bleached corals are actually nearly hypoxic during both the day and the night, and this likely has consequences to their metabolism. So I also measured the rates of photosynthesis and light and dark respiration during the course of the bleaching. And this showed that the gross photosynthesis rate, which we might expect, significantly declined for both species throughout the course of bleaching, which has been shown to be linked to or possibly precede the loss of the symbiodinium. And then also, this was an interesting result that the light respiration and dark respiration rates also significantly declined in the coral host during the course of bleaching, which suggests that their overall respiration rates are being significantly decreased, potentially due to that hypoxia that they're experiencing. And once all of these measurements of oxygen were made, I sampled the corals and I looked at their enzyme activities using the assays that I'd optimized previously. So here we're looking at CS and MDH activities, the aerobic enzymes involved in aerobic pathways, and SDH, our main anaerobic metabolic enzyme. And first of all, we found that bleaching did not cause any significant difference in the aerobic or anaerobic capacity of Agaricea, but there was a clear and significant increase in the metabolic capacity of Orbicella franksi. We found higher but not significantly higher CS activity and significant increases in MDH and SDH. This suggests that there's a species-specific response to bleaching in terms of their ability to up or down regulate metabolic capacity, and that Orbicella franksi might be able to increase metabolic rates to compensate for their loss of symbiodinium, but Agaricea garicides might not. So finally, I did another thermal bleaching experiment using the even more sensitive species, Acropora cervicornis, and I exposed this coral to the same temperature stress, and it took only 4 weeks to bleach. So first of all, right off the bat, we did observe that this species is much more sensitive to bleaching. And I measured enzyme activities in these as well, and found that unlike Agaricea and Orbicella, bleaching resulted in a significant downregulation in the aerobic capacity of Acropora cervicornis, and slight but non-significant decreases in the anaerobic capacity too. So to summarize part 2, I found that thermal stress causes a species-specific difference in both the bleaching rates, which are similar and match those that we observe in the field. But for the first time, we show that bleaching causes species-specific differences in the metabolic capacity of these corals. And the main results were that our fastest bleaching coral had a significant effect, or significant decline in its aerobic capacity as a result of bleaching. Our second most sensitive coral seemed to bleach at rates somewhere in the middle and did not have a significant change in metabolic capacity. And our more robust coral bleached the slowest and was able to increase its anaerobic capacity, which suggests that it might be able to make more energy when it's bleached than these other species. So to bring this up to a more ecological scale, I'd say that these metabolic capacity results actually may underlie our observations of differences in bleaching susceptibility and recovery in these 3 species. So we might start to, or continue to see shifts from these more sensitive coral species to least sensitive ones. And this might be due to their ability to make energy anaerobically when they're bleached. Okay, so I somehow have already reached the end, and I'm really excited to have shared all of that with you. And so in conclusion, I hope that this body of work that I presented conveys not only the importance of understanding basic cellular biology, such as elucidating the anaerobic metabolism of corals, because understanding basic biology lays the foundation for studies of how climate change and other impacts will affect species. In addition, I hope this research conveys the importance of measuring biological changes on different scales of biological organization, because they measure and indicate different things. For example, our protein activity results, our enzyme activity results did not match the results of protein abundance or metabolite abundance, suggesting the scales on which metabolic regulation occurs. And then finally, I was able to optimize and develop some new methods for measuring indicators of coral health, developing enzyme assays and targeted LC-MS techniques. And I hope that these reliable and useful techniques could maybe be used as biomarkers for coral health in future studies on the field, in the reef. And with that, I have a number of people to thank. Thank you all so much for your attention. I'd like to thank my PhD committee, Martine, Dimitri, Peter, Horst, Bill, and Davey. You all have been incredible scientific mentors to me throughout this whole process, and truly each piece of this research could not have been possible without you. I'd like to thank the rest of the Trace Garius lab, Christina, Megan, and Janae, who just defended. Congrats, guys. And the other grad students, Garfield, Dan, Michaela, Angus, and our awesome assistants, Cameron and Shane. The proteomics work wouldn't have been possible without Dr. Lars Tomanek and Marcus Zuzo at Cal Poly SLO. And the metabolomics work was possible because of Gary Suzdak's lab at TSRI and Dr. Yongzheng Su's lab at UCSD. Phil Zarowski has been amazing in helping make all of the aquarium experiments here at Scripps possible. And my Smithsonian Tropical Research Institute was also supported by these three researchers, Dr. Andrew Altieri, Dr. Nancy Knowlton, and Rachel Cullen. And I'd also like to thank the rest of the STRI staff who helped support my field studies there. Here at Scripps, there are so many more people to thank. The Scripps grad department, Gilbert, Denise, the Maureen's, everybody else in the office for making grad life possible, making today possible. Thank you. I started out with a home in CNBC with the NSF Eigert Fellowship, and for that I'd like to thank everyone at CNBC, which is the Center for Marine Biodiversity and Conservation, especially Penny Dockery. My cohort, thank you all. It's been just a pleasure and honor to be part of this with you all. And I got to use some really awesome scientific illustrations from Adi Ken, who's a PhD student here, and as well as Squid Tunes, which is brought to you by Garfield in our lab. And then finally, there are a number of funding sources that supported this work, including 2 NSF grants, the Eigert Fellowship, the Predoctoral Fellowship, department funds, and donor funds. And I know it's trite to end with the sunset, but well, I was really proud of this picture. And I took it right the night before I left for my 6 months of field work in Panama. And that was sort of a pre-introduction to leaving Scripps permanently, because I was met with all of these emotions about leaving this amazing place. Well, then I was lucky enough to come back, but this time I'm not sure how often I'll get to come back. And there are just so many friends and colleagues I have to thank. This is why I didn't put up specific pictures. I knew I'd get emotional, but I just want to thank everyone so much for being part of this journey. I'm so glad we are all there for each other, and I learned a tremendous amount. And thank you so much also to my family for coming out. Thanks.
Awesome job.
Oh, thanks, Martin.
And I just let you to
Yeah, answer
Field the
questions,
questions.
please. Ask away. Abby.
So, first of all, apologies for the cell phone. And
Oh,
second
didn't even
of
notice.
all, I was curious about how bleached corals are very hypoxic. What about cnidarians that never even have zooxanthellae? Are they hypoxic all the time, or?
That's a great question. So our cnidarians that don't have symbiodinium or photosynthesizers, are they constantly hypoxic? And well, one would suspect that they probably would have only net respiration occurring in their tissues, and that this would be drawing down oxygen below ambient seawater conditions. So that's what the bleached coral, it can be used as an analogy to maybe an azooxanthellae cnidarian. However, since the health of the coral is also affected in other ways when they're bleached, I'd say we would need to do additional experiments on unhealthy but non-symbiotic cnidarians. But I would guess that they probably are, because they are only net consuming, have much lower oxygen levels than the surrounding seawater, and therefore maybe they are relying more on anaerobic metabolism. Lisa.
That was great.
Thank you.
So respiration used hypoxia is often accompanied by production of CO2 and hypercapnia. So are the corals taking care of that, or are they also experiencing hypercapnia? That was my first question. The second one is, I know that there's hypoxia has been in the surrounding seawater has been reported increasingly in coral reefs, and is Boca del Toro's one of those, and how might that affect what you
Thank you. Yeah,
found?
both great questions. So the first question was about whether corals might also be experiencing hypercapnia, which is higher CO2 levels which is often concomitant with hypoxia. And while I didn't measure that with CO2 microsensors, there have been other studies that indicate that CO2 levels are higher at night, or in the darkness, I should say, in coral tissues. So this might also have consequences to metabolism, because high CO2 has an acidifying effect. And in fact, it's been demonstrated that corals are actually net dissolving their skeleton, potentially at night, which might be due to higher CO2 levels and also a lower pH. So yeah, so it'll be really interesting to now build upon our individual findings of low oxygen effects on coral metabolism, and now include high CO2 and how the corals might be acid-base regulating in response to lower pH levels. And then the second question was about hypoxia as a growing issue for coral reefs, and is Boca del Toro, where I did the fieldwork, an area that's being affected, if I remember that correctly. And in fact, yes, so in Bocas, there was a recent field study that measured oxygen levels in the bay there. So much of, or Boca del Toro is this island network with this big bay. And there have been studies showing that oxygen levels have been very low, even hypoxic or anoxic at certain depths within that bay. And one of my strike supervisors, Andrew Altieri, did a study, or conducted that study, monitoring hypoxia in Bocas, as well as showing the differential susceptibility of coral species to mortality. And so in fact, I would say that hypoxia is a growing threat for coral reefs. As part of that paper, which they recently published in PNAS, they also did a meta-analysis of all of the studies that mention coral reefs and hypoxia, and find that while this is kind of an underexplored topic. There are more and more cases being reported of low oxygen events occurring on reefs, and drawing attention to the need for more studies of the effects, because already, as my findings show, corals experience nightly hypoxia, bleached corals experience constant hypoxia. What about when you put otherwise healthy corals in conditions of low oxygen all the time? There will probably be consequences to their metabolism as well. So great questions. Any other questions? Yes?
Yeah, I kind of a bunch of questions rattling around, so I apologize if they kind of come out stream conscious. But so the whole presentation was a little confused because you referred to corals. Are you talking about the system of the Symbiodinium and the coral species itself? Like when you did those metabolomic extractions, are you talking about both of them together?
That's a good question, and I was talking about, I did use the whole organism, so both the coral and the Symbiodinium, but I was talking about just the coral fraction that we identified, so the coral-specific metabolic proteins and metabolites. So thank you for helping me clarify that. But we were able to detect Symbiodinium-specific metabolites and proteins as well.
Okay.
And those also did not, those showed a similar pattern to the coral proteins where there weren't huge changes day and night in proteins, but there were more changes for the metabolites.
Okay.
Okay, thanks. Yes?
Thank you for a very nice talk. So your measurements were done in the lab, right? And you measure the concentrations in the boundary layer. How is this boundary layer affected by the flow and the turbulence? And your measurements, how do they apply to a field environment where you usually have more flow, maybe breaking waves and things like that?
Great question. So how is this diffusive boundary layer, or the coral's microenvironment, affected by conditions that you'd experience on a reef, specifically flow, waves, things like that? And yeah, it's true that I had to do my study measurements in a controlled lab environment to eliminate those influences, because the amount of flow is indirectly proportional to the thickness of the boundary layer, so that when you have higher rates of flow, you basically get a washing away of the boundary layer, and so you get a much smaller microenvironment. So you can imagine that on a coral reef, which is constantly experiencing the action of tides and waves, that diffusive boundary layer is probably constantly changing in thickness. And not only from waves and tides, but you also have the effect of the morphology of the coral colonies themselves, where corals on the outside of the colony are probably experiencing those dynamic fluctuations in the diffusive boundary layer much more than corals on the inside of the colony, which are sheltered and having lower flow. So it would be really interesting to use these microelectrodes in situ. And yeah, there is a really cool in situ system that's built for making these sorts of measurements in the field. And maybe during a future postdoc or another PhD student could do that to apply, see what level of variability there is in the field. Thanks. Okay. Yeah. There you go.
In addition to that, if you compare your two species, of course, you probably have very strong differences in the thickness of the DVL versus your branching. You have thin DVL, and your muscle core, you have thick DVL. And also you have in a muscle core much more spatial heterogeneity, right? So for instance, in the sumosoft tissue, you have a thin DVL, and you have a polyp thick. So maybe some of your biological findings, could they be explained by these differences, inherent differences in the thickness of the DVL and subsequently the likelihood for mass transfer mutation?
Yeah, that's an interesting point. So if I understand correctly, the differences in the morphology and the tissue thicknesses of the coral species, whether that's affecting the DVL. And I know there have been some studies done on this very subject that suggest that thicker, more maybe metabolically active tissues might have different diffusive boundary layer thickness than much thinner tissue like species like the Acroporas, which might have a much smaller diffusive boundary layer. And so what was interesting in my findings was that even though I used with different tissue thicknesses and morphologies, and maybe I can go back to that slide, that there was no significant species difference between the Agaricea, which is a very thin tissued species, and Orbicella, much thicker species, in their oxygen concentrations. I did not actually, I don't recall what the diffusive boundary layer thickness was, but at least all of the concentrations in their tissues were about the same, which is somewhat surprising given potential differences due to tissue thickness. I'm not sure if I fully answered that question.
Yeah, I mean, it's surprising to see that, I don't know, like that you, I mean, if the oxygen concentration changes at the surface, either you have differences in metabolic activity, or you have a difference in thickness of the diffuse boundary layer. So if they are similar and somehow there must be a
Right, there might, since, as you're suggesting, diffusive boundary layer conditions are affected by flow and things, which I controlled for so that these two species were in the same conditions. But then what would be the main difference? That would be their metabolism and in addition to their symbiodinium. So this is suggesting that there's some maybe balancing going on where even if one species had different amounts of photosynthesis, which there was a significant species effect, there's a balancing happening so that the actual oxygen concentrations are about the same. Yeah, Amanda.
Didn't that also, sort of in answer to Daniel's too, have a little bit to do with the coral feeding as well? Because, I mean, that would still alter that level, and maybe if you have one that has a thicker tissue layer or a coral that's able to consume a little bit more to supply itself with energy when it's bleached, then that might change that too. So like,
The respiration.
you might see a different level from that.
Yeah, so another consideration between, as Amanda's bringing up, between these species is that corals, they have the ability to feed. And I didn't bring that up since it's not something I explicitly measured, but these corals were both given the ability to feed, they were in filtered seawater, so they didn't actually have the ability to feed significantly. But since acquiring new substrates, not from their symbiodinium, might affect their respiration rates, that could have been something that impacted respiration during the course of bleaching, but I also tried to control for that by using filtered seawater. Okay, thank you.
Okay, now we'll stay with the committee for a few questions and see you.
Should I mention the party? Oh, you already did. Oh, and just an announcement again. We're going to be having a party in Surfside, which is just down the way, starting around 1:30 with tacos and beers, and it'll be going all afternoon, so I hope you guys can drop by. Thank you.
Should be some parking in front. It's only good for you. Okay.
I'll take her outside. It's so hot.
Yeah, it's hot.
It's very stuffy. It's really outside.